So, good evening, everyone. This side Madhvi, your host for 3 days International Faculty Development Program on strengthening research and innovation ecosystem in higher education, which is jointly organized by Rayat Bahara University, Mohali, Just Agriculture Education Group, ISA STR Society, Noida. So, our guest will be joining us shortly, then we will start our sessions. So the first guest of our session will be Dr. Ankit Mangotra. He is the Deputy Director Research, Scots Jammu. So sir will be here within 10 or 5 minutes. Then we will start the session. So, participants, we will be starting our session in just 5 minutes. Actually, the guest is currently occupied with another meeting, but within 5 or 10 minutes, he will be here. Then we will be starting our session shortly after their arrival. But thank you for your patience also. So, just give us 5 to 10 minutes, then we will start the session. Good evening, sir.
Good evening, Madhvi. Uh, I am audible.
Yes, sir, you're audible.
Okay. Can I start directly?
Yes, sir, you can start your
Okay. First of all,
session.
very good evening and very sorry for the delay. So, just a quick question, Madhvi. I just want to know about the participant of in one two line. Just tell me what are the background of participants so that I can.
Sir, it is like mixed audience. They are the working professional also, they are the students also, sir.
Okay, great. So, my screen is visible.
Yes, sir,
Presentation.
it is visible. Yes, sir.
Okay. Okay. Thank you. So, good evening, everyone. Myself, Dr. Ankit Mogotra. So today, I will just tell in brief about the what are the current areas of research in animal genetics and breeding. So I will try to keep my topic very generalized, so that person from different background can also use these techniques in their respective fields. So if somebody will have some any query or question, we will take that after the presentation. So when we are telling about animal genetics and breeding, or we can say especially genetics, in which how we can use different, how we can plan our research, how we can go for what are the different techniques are there in market. Suppose I am a student, okay, so I want to plan one proposal in genetics, in animal and genetics and breeding. So what are the how my guide told me, okay, you have to plan your synopsis work and go for it. So what is my first step I have to go for? So whenever we are planning for research, the things will come in our mind. The first main thing is idea. Idea related what? In which field the research is required. So when we are telling about animals, why we are keeping animals? Mostly, and people are keeping animal for milk production, for meat production, and for wool production, for production related or reproduction related.
So
So as
sorry
we,
to disturb. Can you make it on slideshow?
yeah, I am coming. So is it my slides are moving now?
No, sir, not yet.
Is it moving
Yeah,
now?
now it is moving, sir.
Okay. So when we are telling about different livestock growth, what in our global world, what is the demand? We know that there are different challenges are there, like population growth is increasing. When population growth is increasing, definitely there are issues of food security. We have to meet, we have to eat. Then when we are growing, urbanization is there. There is lack of plants, animals, our diversity, and that lead to climate change. So ultimately, if we have to balance this, how our future generation will survive. So there are so many things will come in our mind. As we know that temperature is increasing, different climates are coming. So in that case, our food chain, whether it is plants or animals, we have to select such animals that are climate resilient. Or we can say nowadays too much outbreaks are there in plants, animals, as well as human, like corona. So how many vaccines we will develop, how many antibiotics we have, and how many antibiotics we can develop. So there are a lot of things are there, that challenges are there that we have to face. So when we are telling about genetics, a last theory is there, we can say survival for the fittest. So what does it mean in genetic sense? The person having good immunity, good genetic component that can survive in the adverse condition. So that point of view, when we are taking, we are telling in animals, there are two main tools that we can use for improvement in animals, or we can breed animals. Suppose मैं मुझे आई वांट कि मुझे एक ऐसी काउ चाहिए जो हाई टेम्परेचर में भी जिसको डिजीज ना हो और वो मिल्क प्रोडक्शन भी कम ना करे. या if I need a particular sheep or goat जो डिफरेंट लेस वो कम खाए और उसका मीट प्रोडक्शन ज्यादा हो. और आई वांट ए पोल्ट्री, ऐसी पोल्ट्री हो जिसको डिजीज ना हो, वायरल डिजीज ना हो. सो हाउ कैन वी गो फॉर दैट? So there are two tools. One is selection, and other one is mating plans. What when we are telling selection in a layman language, I can say, I can explain selection कि हमने किसी को select करना. I have to select a superior animal. Superior in what sense? If I need, suppose there are 5 animals in a particular place, they are eating same feed, they are in same environment, but out of 5, one animal is not getting any disease, 4 animals are getting disease. They are in same environment, safe feeding. Every person is handling the same. So that variation in that particular animal is might be due to genes or due to environment. So as a breeder, we have to first select the individual based on their phenotypic trait. So we can say phenotype. That phenotype of an individual depends upon very two important things. One is genotype, another one is environment. So ultimately, when we are going for selection, किसी भी चीज की हमने सिलेक्शन करनी है, बट वेदर इट इज अ सपोज आई वांट ए प्लांट, आई आई नीड ए प्लांट, ऐसा प्लांट चाहिए जिसको भी डिजीज ना लगे या जिसकी ग्रोथ ज्यादा हो. तो व्हाट फैक्टर्स वी हैव टू कंसीडर, वी हैव टू कंसीडर देयर जीनोटाइप. So we have to plan accordingly whenever we are planning any research in a genetics and biomedical field for disease diagnosis. The first important thing is phenotype. Phenotype means data. First we have, when we have phenotype, then we have to search that the variation in a particular phenotype is due to what, whether it's to genes, whether it is due to DNA, or it is due to mRNA or other factors. So when we are, as this topic is related to animals and livestock, we are mostly consider, when we are considering animals and livestock, we are targeting three traits कि आने वाले टाइम पे, इन कमिंग इन फ्यूचर, इन कमिंग ईयर्स, व्हाट इज अवर मेन फोकस इज लाइक दैट कि जितना भी क्लाइमेट चेंज हो रहा है उनका इफेक्ट प्रोडक्शन पे, reproduction पे और animal health पे. These are the main three targets that directly affect the farmer's pocket. Suppose a particular animal, farmer purchased a cow or buffalo of 1 lakh rupees, and that animal is eating but not giving milk, or that animal is not getting pregnant, or that animal is getting sick day by day. So that affect the farmer's pocket, because 70% of our farmers are from rural economy that depend upon livestock. So in that case, we have, it's our responsibility is a current area of research. We have to select genetically superior animals that will produce more, that will give one, that will get pregnant regularly, and that will be prone to less disease, or we can say that will be more resistant to disease. So when we are going for selection, phenotypic selection means based on their data records, we are either go from parent side, either we will select based on their mother performance, with a father performance, they based on their progeny performance. There are different types of traits. For example, अगर जैसे if somebody is from animal background या किसी रूरल फार्मर्स को बिलोंग करता है, जहाँ हमने +2 में जब पढ़ा था, हमने हम बोलते थे कि मेंडल ने बोला था कि टॉल टॉल प्लांट को स्मॉल स्मॉल प्लांट से क्रॉस करवाएंगे तो जो उसकी प्रोजेनी आएगी टॉल ही आएगी, बिकॉज ही नोज दैट देयर आर जीन्स आर देयर दैट इज डोमिनेंट ओवर अदर जीन. सो हमने अगर पेरेंट्स टॉल सेलेक्ट किए हैं, ह्यूमन भी हम जब हमारे बेबी होता है हम बोलते हैं किस पे शक्ल गई है, इसकी मदर पे है, फादर पे है, इसकी हाइट किस पे गई है. सो दीज रिजेम्बलेंस बिटवीन द progeny or parents that depend upon the genes that will pass on from parent to offspring. सो ये जो रिजेम्बलेंस हम देखते हैं, फर्स्ट ऑफ ऑल हम क्लासीफाई करते हैं कि रिजेम्बलेंस जो 50% इज कमिंग फ्रॉम मदर एंड 50% इज कमिंग फ्रॉम फादर, और हम ऐसे बोल सकते हैं 50% इज कमिंग फ्रॉम पैटर्नल साइड एंड 50% इज कम फ्रॉम maternal side. So whenever we are selecting a particular individual, based on this basis of selection, we can say the performance of a particular individual is due to 50-50 genes that will come from sire as well as from dam. Sire means father, dam means mother in genetic sense. So when we are coming to gene, suppose I can say I have in my body there are 46 chromosomes. So that 46 chromosomes, different types of interactions are there in our body. Like suppose my particular gene, A gene, is affecting B gene, having a dominance effect at a particular locus. Okay, so when meiosis is going on, when my genes will pass on to my progeny, half will go, only half, the combination genotype will never transfer, only genes will pass on. So when we are considering a whole genetic effect, we are considering a fully genetic part. It is not a whole genetic part that it will transfer, it is only the additive part that will transfer from parent to offspring. So how did genomic change the things? Okay, I think everybody, आप में से किसी ने मूवी देखी होगी विकी डोनर. So in that movie there is a very good example of selection of a donor animal. When in human also IVF there is a very common, so many fertility issues are there. So what are the prerequisite when we are selecting a donor animal? There are number of factors are there, like their father performance, mother performance, their height, their semen quality, cells quality, how their DNA works. So likewise in animal and in plants also, we are checking their whole DNA. We are checking, screening their DNA. Or disease to disease, we are checking whether a particular disease is due to what genes. Because if we'll say about humans, we have 46 chromosomes in our cell out, and our whole genome size is about 3 GB. And out in this 43 chromosome, there are more than 28,000 genes are there, approx 28,000 genes. And genes that are present on one chromosome in all in human, if I am telling about particular species, suppose I am having a blood group, other is having a B blood group. The gene of blood group that is present on my particular, suppose present on chromosome 1, same other individual having B blood group, at the same position that particular B allele is present on other chromosome. That is the beauty of nature. So whenever we are going for selection, we are screening genes, and we have to associate their genes, because I told there are more than 28,000 genes are there. Every gene is having different role. There, some genes are major genes, some genes are minor genes. Some are directly affecting the trait, some are in combination affecting the traits. So when we are telling about selection, it is not so easy. We have to take care of so many things. What is the interaction between genes? Whether it is a interallelic interaction or intrallelic interaction, or there are some genes that are affected by environment. Suppose I am taking daily butter parantha, another person is also taking butter parantha. I am getting fatty very easily. Other is not getting fatty because of metabolism or because of genes. My genes are responding more to particular butter parantha compared to other person, because of there is a variation in my gene expression. So there are n number of things are there. Like in particular condition, one animal is getting diseased, other is not getting a disease. It means that due to their weaker immune genes, that animal is more prone to that particular bacteria, virus, or some disease compared to other. So number of things are we have to consider when we are going for genomic selection. So when we are going for genomic selection, as I told, we have to explore the markers that are present on a chromosome at a particular locus, that is at a particular locus, or that markers. There is one term in this slide, that is QTL, that is a quantitative trait loci. In animals, there are two types of traits. One is qualitative, another one is quantitative. Quantitative traits means like milk production. We can say how milk production that can be measured. Milk production, fat production, body weight of sheep and goat. So these are quantitative traits, and these traits are controlled by thousands of genes. So whatever genes, suppose one gene at chromosome number 1 affecting this trait milk production, other at chromosome number 2 affecting the milk production, one other gene at chromosome 1 but at other locus affecting. So all these genes present in different locus affecting a particular trait, that is a milk production. So we can say that these are the quantitative trait loci. So in our genome, there are different types of, there are different locuses are there, where genes are there that are affecting a particular trait. That particular locuses are known as quantitative trait loci. So we have, how we have to detect these locations. So very first important thing, suppose I planned a particular research, I want to explore that in animals one disease is there, mastitis, that is like human it is a breast cancer. In animals we can say it is an inflammation of udder. So suppose I want to check which are the genes are responsible that are affecting this particular trait in animals. So how we will identify these genes? First I have to identify the locus. How we will detect this locus? So first of all, I have to take the blood, and either we have to take the hair roots or we have to take other tissue. We have to take the DNA, isolate the DNA. There are different methods to isolate the DNA. Then, after taking that DNA, we have to, suppose we are taking a single gene. Either we have to go for whole genome by using NGS, or we have to go for a particular immune genes. So we have to screen the genes by using different markers to check. Suppose there are 5 animals that are affected animals, 5 animals that are non-affected animals at a particular place, and our hypothesis is like that. that in 5 animals that are affected and those other 5 animals that are non-affected, there are some markers or genes that lead to variation. Suppose a particular individual is having a gene, other is having a B gene, and we are trying to associate that A gene animals are more prone to disease compared to B gene. So that can be, that marker can, if we identify that marker, that can be used as a diagnostic. फॉर एग्जाम्पल अगर हमने लीवर के टेस्ट करवाने हैं तो एसजीपीटी करवाते हैं जाके. वी आर गिविंग ब्लड, there are two enzymes, they are checking the elevation level of that two enzymes, and they are telling okay, your liver is fatty and all things. Similarly, at genomic level we are trying to identify some markers based on their nucleotide positions or nucleotide variations. We can say them SNPs, that can be used as a diagnostic marker, or we can use for selection purpose, for breeding purpose. So when we are telling about a single marker basis selection, we are either using, there are different kinds of molecular markers techniques we are using. One is PCR RFLP, other one RAPD, AFLP, mini satellite, microsatellite, SNP. Most commonly marker we are using SNP. And what is SNP? For a house, I am just giving one very small example, that is a single nucleotide morphism, a single nucleotide polymorphism. This is a very simple example. Whatever I told right now, I'm trying to sum up in one slide. There are six individuals. There are six individual. Six cattle are there. First three individual I categorize as a low RFI, and last three individual I categorized as a high RFI. RFI means residual feed intake. We can say in animals that animals having low RFI are best for production because they are eating less, producing more. Okay. High RFI, we are telling that animal are low in performance comparative to uh, low RFI. Okay. Now we categorize first three from based on their phenotypic data, their feeding data, milk data. Based on their feeding data, we categorize the first three individuals are low RFI and other three are high RFI. Now our target is to identify a marker in the genome. So a particular gene, we isolated a particular gene and we characterized it. If we'll take first locus, we'll see in first three individual AAA. In fourth individual, in place of A, it is C, but other two is AA. So there is a variation. A to C nucleotide variation is there, SNP is there, but that is not a marker we can say, because in low FRI A is there, in second in high RFI also A allele is there. So we can say it is a marker. In second locus we can say first individual C, in low RFI 1T 2C are there, whereas in high RFI 2T and 1C is there. Here also in both groups both alleles are there, so we can't say it is also a marker. Then third locus we are checking. Similarly, in third locus, both alleles in high also we are getting, low also getting. But in fourth locus, you see in all the three individual at a particular locus, A allele is there, whereas in high it is G is there. So that can be used as a marker. It is not easy to find that, but there are examples. People identified this marker and using in genetics for in disease. There are different diseases are there. There are different production trait and reproduction traits are there. So in that case, we can say in high individual at a particular locus, all are G, whereas in low RFI, in particular individual, only A is there. See, SNP is there in all locus, but which SNP can be used as a marker, that will depend upon our data. That will depend. If suppose हमारे पास ये हमें नहीं पता होता कौन से एनिमल लो आरएफआई हैं कौन से हाई आरएफआई हैं, तो जीनोमिक डेटा से हम कुछ नहीं कर पाएंगे. वी हैव जीनोटिपिक डेटा, बट हम कैसे आइडेंटिफाई कर पाएंगे जब तक हमारे पास फिनोटिपिक डेटा नहीं होगा. So when we are telling genomics is very important, no doubt. Molecular genetics is very important, but unless and until we will not classify the groups based on their phenotype, association will not be possible and we will not come to a particular conclusive results. Hope this slide is justifiable. Uh, any quick question on this slide if somebody didn't got? Any quick question on this slide? Or I will proceed.
So you can proceed if you want, like next 10 to 15
Okay,
minutes.
great. Okay. So Now, when we are telling how we have to identify these markers, so when we are telling about genetics, either in human as well as in plants as well as in animals, there are three techniques are there. Right now, genomics is progressing very fast. One is Sanger sequencing. In Sanger sequencing, what we are doing, small genes we are isolating by using PCR, and then these particular small fragments we are characterizing by using Sanger sequencing. Now there are DNA microarray methods are there. These are chip methods. In a small chip, either it is SNP based or mRNA based panels are there that are used in diagnostic. Illumina and Affymetrix, there are two companies that are providing these chips now in India. NDDB and NDRI, they are and in GBRC, Gujarat, they are doing wonderful job in this area, and they also prepared some marker chips of reported panels of humans as well as in animals. Now, other one is whole genome sequence, that is the NGS platform. See, there is difference between microarray and whole genome. In microarray, a particular slide is there. In that, only a known whatever particular SNPs or particular mRNAs are there that we can identify. But in a whole genome, we can characterize whole genome. We can get novel SNPs also, and whole genome, there is a Lumina platform or nanopore sequencing are there, or Ion Torrent, Thermo Fisher. There are two, three companies are there, like Thermo Fisher, Ion Torrent, that they're giving Ion Torrent. Okay, Illumina, they're giving NGS solutions, and one nanopore, there is that they're also we are using for whole genome sequencing. Now, why we have to use these? For example, in if we are telling about humans, we can say in humans, if you are going to AIMS or you are going to any now good hospital like PGHND or any private hospital in abroad also, there are different panels are there. Likewise, in females, the ovarian cancer and breast cancer is very, now people are very much aware in India now. Earlier it was not like that. So there are panels of genes. They are going there. See, before disease you have to test, we have to aware, we are giving blood, they are simply taking your DNA, and they identified that for breast cancer gene, these are the genes. If there is a mutation in these genes, there are 45 genes panel is there, this one 45 gene breast panel is there. These panel out of 25,000, they identified, they prepared a panel that out of these are the main major genes that are affecting that is related to breast cancer. So they are checking the polymorphism in these genes or expression in these genes, and they are giving you solutions, genetic counseling. Okay, if some individuals having a history background of breast cancer or ovarian cancer, people are now aware. They are not waiting for that. They are going, they are checking if they are carrying that particular allele that lead to cancer in their later stage. They are going for mastectomy or hysterectomy. Similarly, in prostate cancer, there are different identified panels are there in humans. Similar way, in animals, we are exploring these panels. Now, in animals, there are high density chips are available for cattle, sheep, goat, all species are there that can be used to identify such panels that can be used in selection as well for diagnostics. Apart from this, there is now advancement. When you are planning for research, you can select different techniques like CRISPR Cas also. In animals and humans in India it is banned, but in cell lines, in plants and in cell lines, you can check the expression of particular gene, knock out some genes, or you can add some genes, and you can see that in animals, suppose in animals mostly CRISPR Cas is in abroad, people are using for myostatin gene. One particular gene is their myostatin that will leads to double muscles. See, why people are rearing sheep and goat for meat purpose. If a particular farmer is getting a more meat in their sheep, they will earn more money. So there is a myostatin gene polymorphism is there outside India. You will get, if you will search literature, you will get publication also that people are using myostatin gene for increasing doubling muscles in bulls also outside people in outside India. Marker assisted selection is based on single markers that can be used. Now, in genomics, where we are going right now, apart from DNA marker, now there is one more branch is there, metagenomics. Okay, in animals, now global warming is there, and people are blaming that this carbon methane production is more from animals. Okay, now there nowadays there are different startups are there. Recently, I am going through one startup. What they did, they identified some good bacteria, they identified some microbes that lead to reduce reduce this methane production in animals. How they did it? They took the human flora and they'll go for metagenomics. They identified all the microbe species in a particular human, and they identified which are the microbes that lead to high production and which are the microbes that lead to low production. Now they prepared the culture of this bacteria, and they are selling throughout the Europe. They are selling that culture and they are getting too much money. Similarly, nutrigenomics. फॉर एग्जाम्पल आई एम टेकिंग वन ह्यूमन एग्जाम्पल. वट इज न्यूट्रिजोनोमिक्स? जब सभी कोरोना चल रहा था तो सभी बोल रहे थे काढ़ा पियो, या यू टेक जिंजर टी, या यू टेक दिस एंड ऑल दिस. वाई पीपल आर टेलिंग दिस? बिकॉज ऑल दीज आर immunomodulators. These are enhancing your expression of immune genes so that our immunity will get increase. Similar way, in animals, there are some feeds are there. If we are giving that feed, animals are less prone to disease. Their immunity will be stronger. Or there are some feeds, if animals are taking, they are producing more meat, giving more milk. So in the nutrigenomics, we are mostly focusing on mRNA expression of genes, not on SNPs or mutation. So we are trying to identify the genes which are expressing. So if we came to know that if we are feeding berseem to particular cow, or we are feeding moringa to cow, they are less prone to disease and having producing more milk. That is okay. But what actually, how moringa is affecting animals, how it is changing the gene expression, so that we have to identify, so that we can identify that panels, and that can be patent, that can be published. So I'm cut shorting it. For example, this is a very study we conducted in animals. These are beautiful lambs of 3 month old. 3 month body weight is 28 kg. Average body weight in 3 months is 15 to 16 kg. But these animals are having 28 kg. We are just feeding them normal feeding, but we are giving them supplements, immunosupplements, and we want to know what is the effect of these supplements on their gene expression level. So what we did, we two animals we took having lesser body weight, and two animals we took having higher body weight. We go for whole transcriptome analysis, and we at five, see, we can say that more than 5,000 genes that are upregulated, they are having showing more expression in both groups, and more than 6,000 genes are showing less expression. So now, what are the differentially expressed gene in two groups? We came to know that the 28 locus are there, 28 genes are there that are influenced by feeding immunomodulators. Now we can use these panels for publication and for patent. Simply grouping is important. Then we go for RNA isolation, and after RNA isolation, we go for whole transcriptome analysis. And through by using different bioinformatic tools, we identified these are the panels. I am telling in a very layman and a very simple language, but when you are designing study, the prerequisite is that the data collection, your data collection, animal echo data collection, then design of experiment, then sample collection, then after sample collection, your processing of samples, after processing of samples, your analysis part. So many parts are there. Each step lead to error. Now, similarly, in one more study that we identified, where we targeted a particular gene, that is leptin gene. That leptin gene is very important for metabolism in humans and animals. So many, it is related to metabolism. So we identified, and with some SNP in cattle, and we got very good publication of this paper, and we came to know that particular locus and in this leptin gene having showing a mutation, A to G mutation, and this particular AA genotypes is comparatively showing a higher age of first service, but higher lower age of a first calving. Service period is less. So we can say that AA genotype comparatively, even when we try to associate with fertility and reproduction trait, we came to know that AA genotype is less prone to disease compared to GGO genotype. So when we came to know that a particular locus at a particular gene is influencing the trait, what we have to do? As I in the starting slide I told, that selection and mating system. Now we identified a marker. Now what we have to do? In livestock industry, when we are going we are mating, we are planning to mate animals, we will select only those animals or bulls that are having a AA genotype, so that superior progeny will come. As I told you, that genes will transfer from parent to offspring. So this marker can be incorporated in our breeding plans, that can be used for panels so that superior progenies will come. Similarly, in goat also, in goat twinning is very common. Some animals are giving more kids in one time, 2 to 3 kids. One is some are having only one kid. So farmers, what they want that more kids. If they have more kids, they will get more profit. So there are number of genes. These are network of genes. There are number of genes that is affecting the litter size. So similar way, we isolated the DNA. We targeted 4 breeds here. We have their litter size data, and we targeted a particular genomic locus, and we identified a variation at a particular locus, and we came to know that individual with TT genotype is highly superior. This T allele is highly superior comparative to C allele. So we prefer, when we are telling farmers for mating, we will prefer they will give us blood. See, in India, the awareness level in livestock is little less, but in abroad, whenever farmers are going to mate animals or they are planning their mating plans, they are going for their whole genome profiling. They are giving blood to the labs, they are testing and doing that. In India, there is a huge scope. If somebody want to go for startup, in India, there is no animal livestock diagnostic test, genetic testing lab is there. Disease testing labs are there. Lalpath is giving, other one is giving. But for genomic testing lab, except number NDDB and then like Amul, NDDB, GBRC, very less private parties are there. They are doing this genetic marker, this genetic evaluation or characterizing genetic markers in livestock species. So there is a huge scope in this. If somebody want to go for startup, because government is now providing monies for startup also in this budget. In a similar study, here we targeted an immune gene, because in animals, uh, memory infection, memory inflammation is very common, very common. So we identified this a particular marker in MBL in buffalo, especially. This is the first kind of study we identified in buffaloes. In cattle, there are some reported papers are there, and we identified a marker. Not and similarly, see, BRC is breast cancer is a very common marker in human breast cancer. In animals also, we explored this and we got very good findings that we published in a very good journal, that particular variation in this gene also is there that lead to mastitis in animals. So these are the different marker studies, but where we are now, and what we are actually not this much fast compared to developed countries. We are very uh, we can say not up to the level. Why? Now question, why? First thing, we don't have data. I told you, unless and until when somebody we are planning for research, the first thing when you are planning, don't commit hi-fi results if you don't have ground level data. You committed NGS and all things, but when you don't have a phenotypic data, to whom you have to perform NGS, at the end you will come with no fruitful findings. You will not come with any conclusive results. So whatever, whenever we are planning a search, we must know the gaps. In India, the main thing is like that. At field level, farmers are not maintaining data of a particular traits in animals, lack of recording system. So when I am highlighting this, I'm not criticizing it. I'm highlighting this. Why? Because if I want to work on this, I have to meet this challenge. First year, I have for 2 years I have to generate data. I have to generate both phenotypic data as well as DNA data, so that in third or fourth year I can go for some association studies. So these are the main points. uh, we have to incorporate in our study plan, like as I told, like data recording system, genotypic data, genomic data, techniques, their holistic use that we have to use to achieve success in this particular field. Thank you so much.
Thank you so much, sir, for giving your valuable time. So I will be opening the chat box. If anybody have any query, they can interact with sir. So I've opened the
No,
chat box.
There is no question, I think. If somebody want to talk, just let me know. Okay, only if there is no questions, then thank you so much.
Thank you so much, sir, for giving
Okay,
your
wait, one
valuable
message
time.
came. Okay, thank you so much. Thank you, Dr. Smrjit. Thank you so much. Thank you, Madhvi. Can I leave?
Thank you, sir. Yes, sir. Yes, sir.
Thank you. Thank you so much. Thank you.
Okay, so moving forward, I would like to invite our next guest, Professor Dr. Simarjit Kaur, ma'am. So the current position of ma'am is Director International Quality Assurance Cell, Director International Affairs, Dean Students Welfare, Head of Department Life Sciences, Rai Bahara University, Mohali in India. Ma'am has teaching and research experience of overall 21 years, and more than that, she had patents 8, 100 plus research publication and book chapters. She has guided 100 plus PG and UG students. So I welcome you, ma'am, on the behalf of everyone here.
Hello, Madhvi. Am I audible?
Yes, ma'am, you're audible. Mama, are you there?
Yes, yes, Madhvi, I am there. Um,
Okay.
yes, yes. Please allow me to share my screen. I'm unable to share
Just
my screen.
ma'am, you can try now.
No, it's still coming. Host disabled attendee screen sharing. Madhvi,
Yes, ma'am.
still,
I have made you a co-host.
I guess you have made me co-host on the phone. I have taken exit from my mobile phone. Just try to
Okay.
connect to my laptop.
Ma'am, I have made you the co-host from Professor Dr. Simarjit Kaur, I guess.
Yes, 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 yes.
Yes, ma'am, you're the co-host now.
I think there is certain issue. Let me try my mobile phone.
Okay, ma'am. Because from my side, I had made you co-host, ma'am.
Hmm. It is there, but I am unable to share it.
Ma'am, if you want, you can share your PPT with me. I'll share the screen.
Just a second. माधवी, मोबाइल फोन को को होस्ट बना दो. Okay, let me try.
Ma'am, it was visible, but you get disconnected from your phone.
माधवी, मोबाइल फोन से स्क्रीन शेयरिंग मेरी काइंडली ऑन करो.
मैम आपकी स्क्रीन शेयर हुई थी, बट पता नहीं एकदम से आप डिस्कनेक्ट हो गए फोन से.
हाँ, मैं वही कह रही हूँ ना कि इसका दोबारा से मोबाइल फोन का करना.
मैम कर दिया अब.
Just let me know when it is visible.
Just 1 second, ma'am. Yeah, now it is visible, ma'am.
Just a second. I'm feeling like a student today.
Yeah, it's okay, ma'am.
Is it visible, the presentation?
Yes, ma'am. Yes, ma'am. The presentation is visible.
Okay, okay. I think there is some issue with my MacBook. I'm not able to share my screen. So, well, good evening, everyone. Sorry, I kept you waiting for the same. And today I will be discussing about the ethical issues and the scientific misconducts in the research. So, first we need to understand what are the ethics. When we talk about the ethics, that simply means we are talking about some, set of the moral principles or the value. So, what is good in our life, what is bad, what is right, what is wrong, when we are able to differentiate about, these things, then we say we are morally correct, right? So this is something, while we do any kind of the research work, we try to inculcate such kind of values in our research. So when it comes to the research ethic, that means we are talking about the honesty, we are talking about the respect towards the individuals. And who are those individuals? Those are the participants, those are the subjects who are being affected by our research studies or by our reports. So directly or indirectly, whosoever is affected by our, the research work, our research findings, they there we talk about the research ethics. Means we need to be very honest, very respectful, and there are certain guidelines that we need to follow when we talk about the research ethics. Now the next question arises, why there was a need to bring, the ethic codes into the society? A number of examples we are having, the unethical examples, I would say, in the history, especially after the Second World War. There was a lot of chaos going on, and there was a lot of research going on, in fact, I would say. And those were the very, you can say, the inhuman type of experimentation was going on, like the people, the scientists, the researchers, they were checking the breaking and the re-breaking of the bones just to say how many times they could be broken before healing failed to occur. So this was happening at the time of the Nazi, and even the patients, they had been injected with the live cancer cell. And you wouldn't believe, 400 men, they had been left to suffer with syphilis long after a cure. Penicillin was available, but they were not being treated. So what I'm trying to say, a number of unethical experiments were going on. So that was the reason there was a need to bring the ethical code into the form. And the first code that was brought into the form, that was the Nuremberg Code. So this Nuremberg Code brought 10 guidelines to specifically with reference to the human experimentation. So if the human beings are participating in any kind of the study, then this code was implemented, which is known as the Nuremberg Code. So under this code, the first, the most important guideline was to take the consent of the individual. So what happens, sometimes we forcefully, the experimentation was going on, whether I'm willing to share my information or not. So under this code, the subject, they need to give their, the consent for the same. And whatever type of results are there, they should be greater than, they should be in the interest of the society, they should be for the greater good of the society. When we say the benefits, they should be on a higher side. And human experiments, they should be based on the previous animal experimentation. So what was happening at that time period, the animal experimentation was not carried out, and directly humans, they were the subject trial. So under this Nuremberg Code, human experiments, they should be based on the previous animal experimentation only. And it should be conducted by avoiding any kind of physical and the mental suffering or any kind of the injury, only if the subjects are comfortable, and the risk, they should not exceed the benefits. And most importantly, when human subjects are there, that means adequate facilities, they should be used to protect the subjects. And it should be, the experiments, they should be conducted only by the qualified scientist. And still these codes are very much applicable, like for experimentation on the human beings or for human trials, qualified scientists, you need to seek the permission. Even for the animal ethical committee on the animal experimentation, we always seek the permission. And when it comes to the human trial, the qualified scientists or the researchers only are allowed to conduct such kind of studies. And in the favor of the subjects, like they are in the position to withdraw at any time they are in the position to take the exit from your, the experiment. And the scientist who is conducting the experiments, he must be prepared to terminate the experiment if he feels like some kind of injury is there, or disability is there, or death is likely to occur. So at any stage, he should be in the position to terminate the experiment. And after this Nuremberg Code, when it was made clear to, like, these 10 guidelines were shared with the researchers and with the scientists, and after that Belmont report, it came into force 25 years later in 1970. For National Research Act brought this Belmont report into 1979. So this report was based on the three main principles. Like, first is the respect of the person, it should be there. Then beneficence, like it should be for the greater good of the society, and the benefit should be on the higher side, and risk should be minimized, and fairness should be there while selecting the individuals. It's not like that the needy one, the like you are just paying few bucks, you are choosing the subject. So fairness must be there while choosing the subject. So this was the main, the three principles of for the ethical codes, I would say, of the Belmont report. And afterwards, when it came to the publication of the research findings, then the COP came into force, the Committee on the Publication Ethics, which was founded in 1977. So basically, they were the group of the editors, and they were very friendly, and they used to help each other out with their ethical problems. So I would say it was a kind of informal group, a sport group, and still it is working. And while we publish any kind of the our research findings, so the rules or the guidelines of the COPs are always there, and we have to follow those guidelines. Now, the most important things why these the ethical misconducts are happening, and what types of ethical issues or the scientific misconducts are there. So one thing is this: when we say this is happening, so when we say the misconduct is there, sometimes honest error are there. Unknowingly we are doing certain kind of the misconducts because we are unaware of it, we are actually doing it. So basically this is the violation of the standard code of the scholarly conduct or any ethical behavior while we publish any kind of our professional research work. So it could happen at any stage, like any process, while we conduct any kind of the experimental study, or we are directly collecting the information from the subjects, we are recording it during the reviewing of the process, or during the publication, or during writing. At any stage of our research process, if I say, it may happen. And many times it is I would say honest error, so we are not doing it deliberately. So that is the reason we don't understand sometimes this is the violation of the standard code of the scholarly conduct, and that is the reason we need to study it also, right? But many times what happens, we are aware of it, and we feel like it's okay to little bit tweak the information or the data, just to let that data to fit within our hypothesis, what we have assumed. So that is also one of the major reason. So when we talk about the misconduct, it comes under different categories, like fabrication is there, falsification, plagiarism is there. Then definitely during the publication of data, the violation of the authorship, that is also one of the serious type of offense, or a kind of scientific misconduct. Now when we say, actually, this is one of my favorite examples I share with everyone. When the people, they are, or the scholars, they are not able to understand how they would know, like they have been conducting certain kind of the violation, I always cite this one example. I tell them, okay, there is no mice, there is no injection, right? We never did any kind of experimentation in the laboratory, still we are having the results with us. So that is the scientific misconduct. Or somebody else has done the work and we have stolen the work and presented that work as our own work, that is also a kind of misconduct. Now, how often these misconduct are occurring, or I can say this is a kind of fraud also. So 2% it has been found out, like 2% people they are doing such kind of, the frauds or the scientific misconducts have been happening all over the world. And the people they are under a tremendous pressure. Now one scholar is there, the academician is there, researcher is there, scientist is there. They are working in different kind of, the organization, the private or the government organization. So certain they are having certain goals, or the management is putting a pressure on them. Some people they are doing for, for their own promotion, for the academic promotion, for the career advancement. And some people they are having to desire to go ahead to, to be there in the race, or as a front runner, so the people who are working in the, the projects, they are doing it for the grants also. And most importantly, I say the poor research culture in the organizations that also promote the kind of the such the scientific misconduct. So yes, a number of reasons are there why people they have been doing it, because the pressure or publish or perish pressure, that is always there. And yes, academic promotion, that is also one of the major reason. Now let me explain the types of the misconduct. As I have already said, this picture shows a lot about the kind of the misconduct has been happening in and around us. Let me tell you, like whether we are at some workplace or working in a lab, these are the kind of things. If we look around, we will see a lot of things have been happening in the form of the scientific misconduct. Now first I would like to elaborate what is the falsification, especially for the, the beginners. They don't understand what is the falsification, though they have been doing it. They have been doing it. They are falsifying the data, they are manipulating the data. So there are two different things. One is the fabrication and the other is the falsification. If I say the falsification, that means you are having certain kind of the information during, the field work or the experimentation in the laboratory. You got certain kind of data in your hand, but you feel like now this data doesn't fit within your, the hypothesis. So you are making certain kind of, the manipulations or the alteration in that data. That is the falsification, right? And this is the, I would say, the most common type of the scientific misconduct. And in a study of China, 40% of the investigated misconduct cases were the falsification. So you can say maximum time the falsification that is being done by the researchers, by the scientists, they are doing a little bit of, the manipulations. They are tweaking the data, right? So that that data fit within the hypothesis. And this is not only to change the data at the, once we are having the experimental results. This could be at any form, like you are changing the research material, or you are changing the, the values of the equipment. You are doing certain, making adjustments in the spectrophotometer or any kind of, the equipment. You are changing the process because it is not going or it is not aligning with your, the experimental outcome. So you are changing the process also. You are changing or omitting certain kind of data or the results, so that your data is not accurately represented in the research record. So falsification, this is the most common type of the scientific misconduct. And after listening to this, I guess many of you now will get an idea what you have been doing in your, the laboratories or while gathering the information. Then the research falsification, like the people they are changing the, if I say the kind of, when you change certain kind of measurements, when you look into the microscope and you are changing certain kind of, the informations, you are increasing the bandwidth of the certain area and dimming down the other area. So you can change the, the brightness of your microscope, or you are just, multiplying the dots just to show the density on the higher side. So number of examples are there where you are in the position to falsify the information, and the readers they are not able to find it out, like you have falsified the data. Then second is the fabrication. This is like, as I said earlier, there was no mice, there was no injection. Still, we are having the data. So fabrication means creating the information. Like we never went into the laboratory, we never went to collect certain kind of information from the field. We were never in contact with the subjects. Still, we are having the data with ourselves. That means the creation of the new record of the data or the results, right? So this is like, we are completely lying about. We read the papers of the work of the other researchers, and after that, we fabricated or created certain kind of information. So this is the second most common form of the scientific misconduct. And 27% of the cases, out of 22%, they were labeled the fabrication of the data. And many of the researchers, later on, they were found like they have it was found that they have fabricated the information, and many of their, the papers, they were retracted. So I've cited few examples over here, like fabrication cases in the news. Diedrich Staple was very well renowned social psychologist, and he fabricated most of his, the data, and the 58 of the papers, they were retracted. Likewise, we are having the example of the Yoshitaka Fuzzy. He was a Japanese researcher, and you wouldn't believe 180 of his research findings were retracted, because 126 was totally fabricated, like no experimentations was ever done, and the data was totally fabricated. So you see, we are having a number of examples when we feel like, we will never get caught. It's not the case now. A very sophisticated, the softwares are there, who are in the position to detect the fabrication, the falsification. Even if you have done certain kind of, the statistical analysis of the data, so they are able to find out by using those, this statistical technique or the software. So one is in the position to find out like whether the data is, plagiarized or fabricated, or, it is falsified. So we need to be very careful now, because everything is online. Google has given us so much relief, because we just add few keywords and we get the information. So same has been happening in case of, these deliberate error, I would say, why where we are doing it knowingly also. So you may get caught, and you may lose your academic position or any, the research position. So you need to be very careful when you falsify your information, or when you fabricate or create certain kind of the data. Now the most common type, I say, is the plagiarism. This is like we are just stealing the or copying the information of the others, and we are presenting that work as our own work. Nowadays, if I say the students, they are the ones, the scholars who are into this plagiarism. They feel like it is their right. They have taken the information from the Google, but the Google is providing the information who is being, the written by the other authors or the researcher. So without mentioning the actual source, so if I say without giving the credit to the authors of the original work, if you are publishing that data as your own data, that is the plagiarism. Okay, so plagiarism is also of the different types. This has been happening a lot. I would like to share this, the cartoon, you can say, where the student is telling his teacher, what do you mean all my facts are wrong? I copied everything straight off the internet. Now you see, he was so confident, like I have copied each and everything from the Google. So why teacher is telling me I'm wrong, right? This is plagiarism. That's why he got zero, because the data was, it was verbatim copy pasted, so different type of plagiarisms are there. We are having a global plagiarism, where we are entirely taking up somebody else's work and presenting it as our own work, right? And we are not even mentioning the name of that person. That is the global plagiarism. Like if I'm hiring somebody to write certain kind of assignment for me, and if you find a text online and submit as your own work, that is the global plagiarism. And because when we do certain kind of things, I wouldn't say this is an honest error. This means we are doing it deliberately. We know we are stealing the work of the other researchers or the authors. That means we are doing it deliberately. We are lying about the authorship of the work. So this is one of the most serious types of the plagiarism, and it can have serious consequences. So yes, you need to be very careful while we go for this global plagiarism. And then there is a paraphrasing plagiarism. This is also one of the most common type of para plagiarism, where we are just doing a rephrasing, or changing the, without changing the meaning of the lines, we are rephrasing it. Rephrasing or paraphrasing, it is not itself a plagiarism, um, as long as we properly mention or cite the work of the researcher. So we always suggest, okay, you write it in your own words, but don't forget that is not your work. This work doesn't belong to you. This is the work of the other researchers. So there you need to be very careful. You need to cite the source. So paraphrasing or rephrasing a piece of the text in your own words without citation is a common type of plagiarism, because you are presenting that work as your own work, though it doesn't belong to you, right? So it's better you paraphrase or rephrase a piece of text, and then you also don't forget to cite the work of the original or the name of the original author. Then we are having a verbatim plagiarism, like copy paste. We don't put any efforts to change, the lines, or to paraphrase it or rephrase it. So when we directly copy the text from a source and we paste it in, as our own, the document, again without attribution, means we are not giving credit. One, we are not changing or paraphrasing it, right? When we paraphrase the text, we should not it should not change the meaning of the text. Meaning of the text, it should remain the same, right? So we write it in our own words, but again without attribution. If we are doing it without giving the credit to the original source, then we say this is the verbatim type of the plagiarism. So what the researchers are doing, the structure and the majority of the words, they remain same as in the original. And even if what they do, they delete a couple of the words from here and there, where they feel like, okay, it's better to remove the let's remove this one word. So majority of the words, they remain the same when we say verbatim, like copy pasted as such original. Then we are having this next, the mosaic plagiarism. Mosaic plagiarism, nowadays, the scholars, they are becoming very smart. So they are now into this patchwork plagiarism. No doubt, in patchwork, this is also stealing of the work of the other researchers. But yes, here the researchers, they are putting certain kind of efforts. They are referring a number of, rather than gathering the information from one or two research papers, they are gathering the information from the different sources, right? So they are copying the phrases, the passages, or the ideas from the different sources, and then they are putting together all those all those kind of the information to create a new text or a new document. So by gathering the information from the different sources, they created one new document. So that means in patches, like from here there are certain sources. You take one para from this research paper, then second para from two lines from another one. So likewise, from the different sources, you gather the information, and then you are creating one, the new document. And again, without the attribution, you are not giving the credit to the authors. So which means this is a patchwork plagiarism or a mosaic plagiarism, and you feel like you have put a lot of effort, because you have gathered the information from the different sources. But trust me, there are number of softwares now, like the Turnitin is there. So Arkund is there. So these softwares, they can easily detect, the plagiarism is there. Though you have gathered the information from the different sources, but still, uh, you may get caught. Now we are having a self plagiarism, where we are overconfident. We feel like this is my work, I can use it. Yes, this is your work, you can use it. but you cannot use it like once you have submitted that one particular work to earn certain degree, like you have submitted your work to earn a BSc degree or a MSc degree or a PhD degree. So same piece of work you cannot submit to earn, the another degree. So that is the self plagiarism. All right, so you need to avoid this also. If you want to use your own piece of work also, again you have to cite the author, right? And sometimes you need to, if it is a published, then you need to take the permission from the editor. Now, how we can avoid the plagiarism? See, if you are worried about the accidental plagiarism, you can use a plagiarism checker software. Nowadays, the research scholars, they have been using it. There are lots of plagiarism checking software available online, free of cost, are also there. Otherwise, like in the institutionals, the institutes or the organizations, they are having a subscription to Turnitin, Authenticate, or the Arkun. If I say the 100% accuracy is going to be there in case of this type of the software. Now this was the kind of the scientific misconducts that has been happening while carrying out certain kind of the research work, whether we are doing it in the lab or in the field or gathering certain kind of information by connecting with the subjects. But what happens once we are having the things with us and then we want to publish our work with the in the journals or in the form of a thesis or in the form of certain kind of the write-ups, then we say the yes, the number redundant publication, that is also one of, I would say, the kind of misconduct where we say the duplicate publications are there, simultaneous submissions are there. So as a researcher, this we have been doing a lot. While the redundant publication is the publication of a paper that is substantially similar to a published paper by the same author. So I have already published a paper and I am publishing it again. That is a duplicate publication. And I have submitted my research paper for publication in one of the research journal. And again, the same research paper I have been submitting for publication in the another research journal. That is the simultaneous submission. So in both the cases, it is like the ethical committee can take action against us. So this is not right, right? So whether it's a duplicate publication or simultaneous publication or submission, we should avoid it, because sometimes the researchers, they're under a lot of pressure. They want a very quick publication, so they feel like, okay, they're having a fear of like their paper might get rejected. So considering that, they submit the same work to two or three the research journals. So we should avoid it, right? Or if we have submitted our the paper to one research journal and we want to submit it to the another also, then for the editor we should give in submission like this paper has already been submitted for publication in the another research journal, because it is a wastage of time on the part of, because the paper goes for the peer review process. So it is the waste of the time of the editorial board members, like they are putting your research into the a long process, and suddenly when you get notification from one of the research journal, your paper got accepted and you are withdrawing it from the another also, so another research journal. So that's why this is not allowed. Then there is the overlapping publication, and this many times the researchers they are unaware of the overlapping publication. See, you have published your work and now you are publishing it again. I mean, you have made certain kind of changes. Like if I say you are asking the same question with the different data set, or different question with the same data set, so here and there you are making certain kind of changes, and 40 to 50% of your findings remain the same. That is the overlapping publication. And the research journals, they don't accept your publication if it is an overlapping publication. In few research journal, they allow to have 5 to 10% of the overlapping publication. Otherwise, overlapping of the publication, that is not allowed, because you have been publishing your the same data again and again by making few changes, like you are changing the title or you are changing the objective, and your findings are almost the same, and with the you're ending up with the similar kind of the the outcome. So now the question here arises, can we publish same data twice? No, we cannot publish the same data twice, especially if I say if our hypothesis, raw data, main study findings, conclusion jo hai, if they are the same, then we would say we cannot publish it. That is the duplicate publication, so this is not allowed. And see, the many research scholars, they have been doing it, and at the end one of the paper get retracted. So it's better to avoid humiliation, we should avoid it. Now the second question arises, can I use my published paper in my thesis? Because this is a very common question asked by the scholars. See, it is perfectly acceptable to use one of your previously published paper as your thesis paper. This we simply say is the cumulative thesis, right? So you can use it, but to avoid any kind of the copyright issue, because while we submit or publish a research paper, we submit copyright form also. That means we are giving all the rights to the editor, right? So you should take the permission from the journal where your paper is published, and you can use that published paper as a part of your thesis. Now the next question arises, like, why duplicate publication is a problem? See, it is a republication of a paper without permission. Sometimes when you write to the editorial board and you tell them, okay, this was published at a national level and I now want to publish it to the international level, and you get the permission from the editors, right? Otherwise it violates the copyright, as in most of the cases, as I said earlier, copyright for the paper lies with the journal, not with the authors, right? So we are transferring all the rights during publication to the research journal. So authors are not free to republish their own paper, own work, right? Because we have given our rights to the editors. So before a republication of the paper, you need to seek their permission. And if they don't allow you, you cannot publish it, right? I've cited one case study also, because the scholars, they get confused, like, you have published an article in the regional, at a university level. Suppose university is having its own research journal and you have published your article in that research journal. Now the author submits the paper for the publication to the journal based in some other country, right? So you now you're feeling like, okay, you have published your research work at the local university, now you're want to publish it to the USA or to any other country journal. So would this be considered as a case of duplicate publication? Yes, because your research paper is already published, right? So for this, first you need to inform the second journal, author of the second editor of the second journal of already published paper in the university journal. So you have to give the undertaking, this paper is already published in this the state level or the national level research journal. So, and at the same time, there you give the declaration to where you are submitting your the paper for the republication, right? And to the journal where it is already published, again you need to take the permission from the previous research journal to publish the findings again. And there you also need to justify why you have been doing it. You need to justify, yes, you have been doing it for the greater good of the society, because these are the findings which are published at the state or the national level. Now I want my findings to become the international, right? Now next, the most common kind of the uh, ethical issue or the misconduct our authors they do, this is the slummy slicing. Slummy, it is a piece, very thin piece of meat, so it means what you are doing, you can see in this picture, this is one piece of meat and three dogs are waiting. That means we have to chop it into the three, the different, very thin slices. So slummy slicing are the very thin slicing. So when we say in the research we are doing a slummy slicing, that means we are breaking down a large piece of our research work into the smaller parts or into the smaller portions. If I say we have worked on certain parameters and feel like, okay, why to publish this whole data in the form of a one paper? Why not to get 2 or 3 papers out of this data? That is not, the right thing to do. See, if we are in the position to cut or the convert that data into the 3 papers, only then we should go ahead. Otherwise, if we are converting it or dividing it into a slices, right? So I would say each of which is a least publishable unit, means it makes no sense, right? So our 3 papers I have published, but none of it is making any sense, because those that data was meant to put together, right? So many times the authors, they are doing it to increase their publication count, to get more recognition, and to achieve faster career advancement. Or they feel like, okay, if our publications number will be on higher side, then we will get the more grant for the research projects. So this slummy slicing, this is also one of the common. I have already citing where as a researcher you are thinking, oh my god, I need to publish more papers soon, so maybe I should divide my original study into the smaller parts and publish them separately. Like I have worked on, I rather than publishing it in the form of a one research paper, I am making 3 papers out of it. Okay, so as a researcher, this is honest error, and you feel like, okay, this is the right thing to do. This is my data. I know how to divide it. But see that there you need to see whether it is making any sense or not, or it is a least publishable unit. Like it is not making any kind of sense. You may get it published, but as a reader, I wouldn't be able to make out anything. I have to study 2 or 3 more papers. And sometimes, yes, when it comes to the simultaneous publication, you feel like, oh my god, it would take a long to hear from the journal, so let me submit my paper into the 2 journals at once, so it will save my time. And you feel like, okay, kahin na kahin se to I will receive some, the positive response. So this is also a kind of, the thinking going on in your mind as a researcher, as an author of your paper. So how we could publish our work, ethically? See, if your study is very large and you feel like, okay, you have addressed many distinct questions, right? Then you could reanalyze your results, and you feel like, okay, it from a completely different angle, like same thing with the different perspectives. If you are taking it or given a completely different treatment, then you need to, break down your paper into the smaller parts only then. Or avoid breaking up data unless the study demands it. Okay, just to get 1 or 2 more publication, don't do it. Then focus on the quality of the publication, not on the quantity, because if quality of the paper is very good, then you will be get more citations and your H index will be on the higher side. And if you're using data from the articles that you have previously published, make sure you cite them in the paper. If it is your paper, then again it will fall under the category of the self plagiarism. Anyhow, in any case, you need to give the credit to the authors. And in case if you feel there is an overlap, make sure you declare this is at the time of the submission, because overlapping is might be considered as a duplicate publication, right? If it is more than 40%. So many of the research journal, the Scopus or the UGC care, they don't allow this, the overlapping publication. So if it is a overlapping publication and you are in the position to convince the, the editorial board member why this is so much important, to carry forward that information to justify your final finding, then you need to submit the copies of your potentially overlapping studies to the editorial board also, right? And if you have submitted the same paper to the another journal, then you need to inform the journal editors about it. Okay, so they know that you don't have the intent to deceive them, and you are doing it because you are in the need of publishing your research paper, and anyhow you are hoping one of the research journal might be responding back or accepting your paper for the publication. But yes, you don't have the intent to deceive them, and for that you need to give this declaration to the editorial board while you submit your research paper. Now I have given the examples of like when your paper get retracted, like you have the certain scientific misconducts are there, you have fabricated the information, or you have falsified the data, or plagiarism is there. You can see the paper from the Scopus or the elsewhere are being retracted. So this type of appears on the Google when the papers are retracted. I always cite this example of the Hungarian president, Mr. Paul Smith. He was the Hungarian president, and he has to resign from his position because 20 years back he did some kind of the research work for his PhD thesis. He worked on Olympics medalist, gold medalist, and later on the players, the sports persons filed a complaint against Mr. Paul Smith, like while he was the president of the Hungary, like he has stolen the data of the the other researchers. He has stolen the findings, so he has to resign from his position. So no matter like you are sitting at some very good position in the academics or in the research, once plagiarism is detected, you can be removed from any of your position. So you need to, that is the reason you need to get aware about the the ethical the issues or the scientific misconducts, or there you cannot avoid by saying okay, this is an honest error, because if it is an honest error, we can see it very clearly. And many times when you are very renowned researcher or a scientist, there we can see it cannot be honest error. Means you have been doing it deliberately, you have been doing it to gain certain kind of the benefits. So you need to be very very careful while from conception of the idea to experimentation to the analysis and to the publication of your research work. At each and every stage, uh, wherever you fabricate certain kind of data, or misrepresentation of the data is there, so your research work can be retracted at any stage, right? Even after 20 or 30 years of your the career. And I think we don't want this to happen just because we were into certain kind of the malpractices. So let me have a summary of this, the ethical issues. See, we always say unethical research and writing practices are wrong and it must be avoided. And there is no excuse for the unethical behavior, right? In our real life, we keep talking about the ethics, the moral values, the moral principles. So unethical practices, they always lead to major punishment and embarrassment sooner or later, and withdrawal from your academic or the research position. So take home message to my dear participants or attendees: honesty is the best policy everywhere. uh, बचपन से सुनते आते हैं, right? And this is a fact, so we should have this thing in our minds also. And as a researcher, research integrity, honesty, and respect to the participants or to the to your own studies, that is very very much important. And for that also, if you're working in a group, I always tell you need to choose the honest colleagues also and the seniors for the research and the writing, because the whistleblowers, they are the ones who are always work in your close proximity, right? And the people, like you are working in a group and you don't know, like the other two or three persons, they are fabricating the data or they are not being honest, you are being honest, so as a part of group you could get punished for their the errors. So you need to be very very careful while you choose your colleagues, and you need to plan ahead. Don't rush like the research process or the blueprints of your research, that those should be very well prepared, and so that you could execute it in a well planned manner, and definitely you will get the good results. And you need not to at that then you will feel like, oh, there is no need to manipulate or fabricate the data. So share whatever you learn, right? Sharing doesn't hurt, and always lead by the example, right? So I would say ethical issues are very common nowadays, and misconducts have been happening. It has been happening for a long time, still has been happening. So why the various ethical codes they came into existence to avoid the unethical kind of the experimentation. So those experimentation were being carried out on the human beings. Now what we have been doing, if we are fabricating any kind of data, any kind of information, so what type of findings we are sharing with the society? So that is the reason, still we don't have answer or a cure for HIV AIDS or for cancer. 100% results we are not getting, but because somehow at one or other stage there is a some kind of falsification of the data, misrepresentation of the information. Until unless we choose the ethical guidelines and we are being very honest while doing the kind of the experimentation and publishing the information, and till it reaches the audience, to the readers, and they are in the position to understand it correctly, only if we have shared the information correctly, right? Only then they could take our research work forward in a forward direction, and we would be able to find out certain kind of, the answers to the problems which are still unsolved. Okay, so I always say we need to be very, very honest while we carry any kind of research work. So thank you so very much. At the end, I would like to say, live your life by ethics and publish your work ethically. Thank you so very much. If you are, and I'm extremely sorry I had to use my, the mobile phone for the this, and I was not in the position to share through my MacBook. I'm extremely sorry
I mean,
for.
it's completely okay. It's just you give us your time and share your valuable insights with us, ma'am.
So if anybody is having any questions, please feel free to ask me. Any questions related with the publications?
I can see how can they thanking you in the comment section, ma'am.
Yes, thank you. Thank you so very much for bearing me today. Because
It's okay.
मुझे खुद अच्छा नहीं लग रहा है कि आज थोड़ा सा वो हो गया. But anyways, nevertheless, I'm always available. If any kind of information is required, just don't hesitate to reach me out.
So thank you so much, ma'am, on the behalf of everyone for your inspiring contribution, ma'am.
Thank you so very much, Madhvi. And if any questions are there, you could ask me later on also. Is there
Sure, ma'am, sure.
another session afterwards?
Yes, ma'am, there is one more session.
Okay, okay. So I take leave now. Thank you so very much. Thank you, everyone. God bless you all.
Thank you, ma'am. So, participants, moving further, now I would like to invite our next esteemed guest, Dr. Vandana Thakur, ma'am, Assistant Professor, Department of Horticulture, School of Agriculture, Lovely Professional University. So, I welcome you, ma'am, on the behalf of everyone here.
Good evening, ma'am, and thank you.
Ma'am, you can start with the session.
Okay, ma'am. Ma'am, slides are visible to you or not?
Yes, ma'am, it is visible.
Okay. Good evening, everyone. My name is Dr. Vandana, and today we are going to discuss about the some very recent or kind of a topic that is current area of research in vegetable production. As I have a specialization in a vegetable production, so we are going to discuss a broad area in which we can focus further for the research and development purpose. So first of all, we are going to have some kind of broad area that we have as we are seeing it as a challenge. So when we are going to have an overview on the vegetable production, vegetables are considered to be a herbaceous kind of a plants where we can grow it, and it is going to be available as a cheap, and it is a very good source of the quality minerals, vitamins, and other sources. So once we are going for the production technology of it, we are facing a common challenges that first of all that related to the challenge is the modern vegetable farming that directly includes with the pest, soil degradation, and the climate change. Then rest of the other, we have the other challenge that is to availability of the quality material in context to the yield quality and the sustainability of the varieties that can be more to be available in the market, as we have the lots of the institute that are working on some kind of the problems of it. The first problem is that is climate resilient vegetable cultivars. So once we are talking about the climate resilience, so we have a numbers of abiotic stresses on which we are focusing on it. See, for example, we have a heat, drought, and flood tolerant vegetable that we have to go for the development of it as through by approaching of the traditional breeding method or the genetic modification. When we are talking about these kind of abiotic stresses, we have other part of it, like we have a two category of stresses like biotic and abiotic. Once we are talking about the biotic stresses, that majorly focusing on the pest and the diseases, as it also going to be comparable with the abiotic stresses. And by increasing the population day by day. Hello,
Yeah, in between there is some internet connection.
just have to check, ma'am.
Yeah, now you're audible, ma'am. Now you're clearly audible.
Okay. S- Ma'am, slides are visible now. So the second thing that what we have is precision agriculture and the smart farming. Here, what we are going to discuss, and the second major area is precision farming. Why we are going for the precision farming and the smart farming? Because when we are going for the traditional kind of a methods, we are opting for the kind of the approaches which is very traditional towards the soil health. We are improving it, but with those traditional methods like the organic farming, then the natural farming we are opting, we also have a real time data which we are collecting in terms of the research towards the vegetables that we have. For example, we have a tomato, then we have other kind of a crops like lettuce, then parsley, celery, that we can also grow within the indoor system cultivation, that we'll also call it like hydroponics. Now you can grow it in the protected cultivation, we can go for the aeroponics. Then what we have to do, in the precision farming, we are also using the artificial intelligence in the collaboration of the real time data to monitor the quality and the checkup on the vegetable production. Now precision farming also included some kind of irrigation management, you can do nutrient management, then pest control can be done by using the sensors and the drone. These drones basically are used for the example of the application of your fertilizers. Okay? And we can also apply some kind of the pesticides on the accurate time on the basis of a real time data for the precise location. As we are collecting, first of we have to collect the data for that particular region, and we also have to monitor the health status of that soil, and then we are going to plan further for the requirement of the particular crop and the nutrient management for the same. Then we have some kind of benefits of the precision farming and the smart farming, as we are increasing the productivity. Due to the urbanization, we have a very limited kind of a area where we have to grow a crop for a large population. So what we have to do, we are also moving towards the vertical farming. We can also use some kind of a area where you are using the vertical space of your limited land, and this is also going to be help in the precision farming and the smart farming. Now the third objective. is a sustainable soil and water management. Now, this is the main agenda as with the abiotic stresses, we are also going to have heavy metal stresses that also in the context of soil and water management. In a soil, we are also depleting some kind of a essential nutrient that is available on the surface of the soil. Due to the drip irrigation, sometimes we also have a lot amount of the salinity that is on the top of the soil, and due to which the leaching of the nutrient are is there. Then water management, where we have a very limited source of the water, and the water table is also going to be reduces, and the availability is also going to be reduced day by day. So what we have to do, we are in a going through a sustainable approach where we are going to collaborate with water management and the soil. For example, we are doing this with the using of bio fertilizers. Then we are using the organic fertilizers, biochars. Cover crops are also important for it, and then same we are doing with the mulching practices. And mulching, maybe we have two types of a mulching that we are using. That is, first one is your the organic mulches, and another one is your the type of synthetic mulches we are using. ठीक है, तो organic mulches में that also going to have, and it also helps to add on the nutrients to your soil. But in context of the plastic mulch or the synthetic mulch that we are using, it too is going to reflect some kind of a light. So what is, for example, we have a silver type of a mulching that also act as a reflectance and repellent to the insects. Okay, and then next thing, what is biochar? So biochar is basically we are also managing paddy straws and some kind of a straw, sugarcane straw, that is used for making the biochars, and this is also used as a alternative of a organic nutrient management. Side by side, the cover crops, this is a very kind of a healthy or kind of a organic alternative towards the synthetic fertilizers, so we can use to improvise the soil fertility and flora and fauna on the same time. Now, once we are using the cover crops in the some kind of biofertilizer, for example, FIM, vermicompost, and compost, then it also helps to improvise the soil fertility, and plus it also helps to maintain the moisture level within the soil and the temperature of the soil. And it is also helps to improvise the water efficiency and the irrigation, as we are also using some kind of a irrigation technique like drip irrigation, that is mostly utilized where we have a very less water level. And this is also going to help you to utilize the proper availability of the water towards the plant, and as per the need you are going to apply that water. Okay, then innovation in the water waste management and it use for the vegetable farming, you can do side by side for it. Then we have a third major objective, that is integrated pest management. Okay, as I am talking about the biotic and abiotic stress previously, so abiotic stress may we have the stresses which is related to the environmental factor. For example, we have temperature that is high and low temperature based stresses. Then we have heavy metal stresses, nutrient basis stresses we have. Then we have the other part of it, that is biotic stresses, that is basically related to the pests and the diseases that comes with the living kind of organism, and abiotic stress will come with the environmental factor. Now, we have to go through the integrating approach to manage the pest. For example, if we are relying on 100% on the synthetic pesticide application, so it is also going to affect your soil health, and also it is very hazardous towards the health of the humans. So what we have to do after that, if we are approaching towards the integrated management strategies, so we are relying 80% on the management that is based on the kind of bioformulations you are using, some kind of a microorganism you are using, along with the kind of synthetic pests. So that is kind of integrated, that is going to reduce the hazardous effects of the synthetic pesticides. So in case of the biological control agents, we can use some kind of insects. For example, we have a ladybird beetle that is also called as a natural predator. for the aphids and other sucking pests. Then we have a trichoderma, then we have a trichogramma that is also used as an alternative. And once we are talking about some kind of the crops that also act as a kind of a repellent towards the some nematode. For example, marigold is the best example of it, as you can use as to control the nematodes over there. Then another one we can also use, that is your neem oils and neem kernel based extract are mostly utilized as an organic alternative to manage the pest, whether it's a sucking pest or the chewing kind of a pest. Okay. And the second thing that the varieties, vegetable varieties also have a resistance and a tolerance towards the pesticides and towards the pest. For example, tomato may we have fruit and shoot borer. Then we have brinjal may shoot and fruit borer of brinjal. Then another one we have, at the same time we also have a attack of downy mildew, powdery mildew over there. And we have a number of varieties towards the management of it, and it is very important to develop a new varieties, as after 8 to 10 years, if you are developing a variety that is going to break down the resistance, so continuously we are working on it. Then third one is the digital pest monitoring as AI based early warning systems. So first of all, digital monitoring, uh, pest monitoring and the AI based early warning systems are working as a part of the precision farming, where we are collecting a real-time data of how the life cycle of the pest is going to affect the plants and what part of the plants is going to be affected by it. Then we are going to decide what kind of the management practices we are going to apply on it. Then fifth, current research area, which is very, very important, as we have a very limited land due to the urbanization and the industrialization. So we basically utilizing a very limited area to get the maximum protection from it. And this is emerging as a very new topic, that is vertical farming and the urban agriculture. As everyone know, urban agriculture is related to where we are producing, we are processing, and we are making the vegetables available to the semi-urban and the urban area. And it have some kind of a benefits where we are not able to get the vegetables. So we are also having some concept like truck gardening we are doing over there. Then you can also going for the floating gardening, that is also going to help to grow the vegetables. And the vertical farming is basically we are utilizing the vertical space in terms of the crops which we can select on the basis of their high value crop and with the high prices. In context of the vegetables, we have some kind of a vegetables that we can grow throughout the years, like tomato, cucumber, then lettuce, then parsley, parsnip, and we also have some kind of a medicinal plants towards it, where you are going to get a higher pricing in context of a vertical farming. So there is one kind of a question everyone is going to ask, and most of the time student will ask to us that vertical farming, why we are choosing a crops which have a high rates and the high prices and the high value. See, once we are going for the vertical farming and some kind of the hydroponics, aeroponics, we are opting and aquaponics, because we have a very limited kind of a space and we have to get a maximum yield from it, because these kind of the techniques are basically developed to get 10 times more yield from the limited area. So that's why we are going for the crops which have tiny in growth and have a high rate in our markets. So we are going to get all the investment in terms of a profit to us. Now, vertical farming may that depends upon the structure and the kind of a region you have, and the crops you can select accordingly to the market. Now, basically, we are focusing on the research on hydroponics, aeroponics, and aquaponics for the uh, semi-urban kind of a area where we are targeting for it. And we also have another concept in a vertical farming, or you can say in a vegetable production, that is your vegetable grafting. Vegetable grafting is basically done when we don't have the kind of a resistant variety or the tolerant variety for a particular area, where we are using those susceptible varieties with the resistant root stocks. So you are going to get a quality yield in terms of the sign if it have a quality traits in it, and you have a resistant rootstock that is going to help your susceptible sign to be grown over there properly. And now these vegetable grafting is a kind of the practices which is not a kind of replacement of the breeding or the biotechnological tools, but it is a kind of a approach that can going to help you to use as an alternative technique once, because your varieties are going to take a time for 8 to 10 years to be developed for a new ones. So in during that period, we can use those susceptible variety along with some kind of resistant rootstock to be developed over there. Then another thing is the vertical farming and urban agriculture is some kind of a very expensive approaches where you have to invest somewhat more in an initial stage, and once you are going to manage it, you are going to manage benefits from it and in terms of your revenue. And we have some kind of the impact of light controlled environment we need for it. Okay. Then we are some have examples that you can grow as a indoor farming. Indoor farming you can do rooftop greenhouse cultivation. You can do for your for the consumption purpose as on the basis of your daily needs consumption. Then we have a other research area that is your post-harvest management and the supply chain innovations. Now, as we know that vegetables and fruits have more than 20% or more than of that losses just because we don't have a proper post-harvest chain and the management system for it. And every year we have a lot and a huge amount of losses are just because of these post-harvest management strategy, because we don't have a supply chain, we don't have a cold stored storage, and that is not available in every region with any kind of a state, and we need more investment towards it and also some kind of a government policies. So what we are trying to do as a researcher, we are also working with the vegetable production with the development of new varieties in from traditional method by opting for the breeding. And second thing what we are doing, that is we are also opting for the biotechnology. But there is one aspect of a post harvest management, as we have to focus on extending the shelf life of the product, so we also have to jump on the post harvest management with the collaboration of the food technologies. Now we have some kind of the research that can be extended to enhance the shelf life, for example, with the basis of your packaging material, cold storage, what kind of a temperature you are going to keep for it. Then we have a blockchain and AI application, so you will going to get a better logistic and can reduce the management as you are going to have a real-time data, as I have previously said in the precision farming, because this is also considered to be a part of a precision agriculture where we are managing our post-harvest losses. Now, there is another thing that is very, very kind of the eye-catching kind of a concept, that is the nanotechnology. Nanotechnology is also going to be used in a post-harvest management, as we are using a double coatings for preservation purpose and to maintain the freshness of the vegetables as well as the fruits and the other crops. Now, seventh important point is now, as we know that we have a very limited land and most of the land is heavily contaminated with the heavy metals. So everyone wants to have a healthy alternative of the vegetables and the fruit as well. So we are try to rely on 80% and the more than of 80% to be go on a organic and regenerative agriculture. So I have used two terms over here, organic and regenerative of the agriculture. Organic, everyone knows that we are using or we are opting for the kind of the practices where we are conserving our soil for 3 to 5 years to ensure that so it don't have any kind of the application of synthetic fertilizer, synthetic chemicals, and the GMO crops are not used in organic agriculture, and we are using all the farm based kind of the composting and the material to maintain the soil health, flora and fauna. And it also helping to maintain the yield and without compromising the quality of it. Now, the second term which I have used, that is regenerative agriculture. So, if we'll go in the depth of the regenerative agriculture, this is also a kind of approach where we are opting for the methods to maintain the soil health, the ecosystem of the soil, along with opting for the organic and the kind of healthy practices in which we are using biofertilizer, biostimulants, then we are using the organic alternatives to maintain and to improvise the health of the soil. Now, we are also, as we are going for the studies of it, if we are talking about the research area, now see, every vegetable crop have their own kind of production technology and package of practices, and every crop have their standardization recommended dose of fertilizer. So if we have to go for organic and the production of organic vegetables or regenerative agriculture, so we have to uh, analyze, or we have to go for the studies where we are going to maintain the ratio of 80-20% of the organic and synthetic fertilizer application. For that, we have to go for the continuously studying the kind of what kind of the new microflora, microorganism we can use as an alternative to fix the nitrogen for fulfilling the availability of phosphorus, potassium, and other kind of a mineral content like magnesium, zinc, and calcium. If we are talking about the organic fertilizer, so the best alternative what we are using, or the most of we are trying to use, that is your FIM, then compost, then we are using vermicompost, then we are using for vermi wash. And apart from that, the cover crops may we are mostly rely to have to grow the crops like which helps to fix the nitrogen, and we have a very best alternative for it, that is your legume crops, because legume crops have the ability to fix the atmospheric nitrogen to the soil, and it will improvise the level of the nitrogen in it. Now, if we are going for the other alternative of it, if I am also going to add nitrogen, phosphorus, and potassium, as same with the compost and the vermicompost. Now, as I have told you that we are using three types of the organic fertilizers that fulfill the nutrient requirement, that is your FIM, then we have compost and vermicompost. And these three have a different levels, and it is not a same kind of a thing. FIM is basically a farm yard manure that is going to be a decomposed type of the organic material, that is a product of the cow dung or the any kind of a dung we are using, and then it's going to be decomposed by the bacteria. Now, we have the second one, that is your compost. The compost is prepared by the use of all the agriculture waste we are going to get together. Then we are going to add some kind of a cow dung and cow urine, and cow urine itself has some kind of a bacteria that helps to decompose it. Now, third, what I have said, the term is vermicompost. Vermicompost is basically a kind of the produce which is going to be uh, means to be produced or to be prepared with the help of the worms we'll use for it. For example, we'll mostly use red worms, and those red worms is going to digest that raw material, and it's going to decompose it within the time, and you are going to have a very fine textured kind of the vermicompost, and it is very healthy for the plants, and it is also adds very good amount of nitrogen, phosphorus, and potassium. Apart from these basic kind of your organic alternatives, we can also use biofertilizers, like we can also use biostimulants where we have a very good examples of humic acid, then we can use fulvic acid. And now in today's researcher are also using like seaweed extract, then you can also use some kind chitin, and then based extract can be used for it. Now, if we'll go for enhancing the biodiversity and the microbial health, as we are using this all organic alternative, it not only going to maintain the soil fertility, but also going to improvise the some kind of the biodiversity and microbial health too. Now, the another thing where we are comparing the organic versus conventional vegetable farming in context to the impact of the yield. See, as I told you that biostimulants are also helps to manage the biotic and abiotic stresses. If we are talking about the conventional vegetable farming, here we are using some kind of the synthetic fertilizer and we are more relying on it. So if we are focusing on the organic ones, so we are reducing the doses of it, and then we are going to have 100% healthy crop with it. Now we have a eighth objective, that is, as I told you, that biotechnology and the functional food. So functional foods may basically, if you are developing some kind of a functional food or medicinal benefits of it in context of biotechnology may see we are doing some kind of a technology which is advanced like talent, zinc, then we are using CRISPR-Cas. As CRISPR, everyone know this is a gene editing technique where you can going to improvise or do repeat or delete some kind of a genes or the kind of the part of the DNA, and you can introduce a new gene to it, so it is going to have improved character you can introduce in it. Genetic engineering basically helps to enhance the nutritional content of vegetables. We have a very good example of the crops like tomato may we have genetically induced kind of a varieties. Then we have, that is the example is flavor saver, that is genetically modified kind of a crop. And then we have the another option of a biofortification of your cauliflower, and that is developed through breeding approach, that is not from the biotechnology, as that crop have a variety, that is your Pusa beta kesari 1, which is a very good source of antioxidant, and it is yellow in color. We also have purple color cauliflower, then we also have a red cabbage which have an anthocyanin in it, and you can consume it as a healthy alternative as a salad. You can just cook it. Then we have another options of, as we have radish. Then another one what we have, like we have sweet potatoes which is have a color in it, anthocyanin pigment, and it is very good in antioxidant activities. Now, another concept of it, as I'm discussing about nanotechnology.
Ma'am, you are not audible.
Hello.
Yes, ma'am, now you're audible.
Okay. So, as I'm discussing about nanotechnology, we have uh, some kind of the alternative, as we are discussing about organic. Then we have focusing on the research area of the vertical farming. Then we are discussed about hydroponic, aeroponic. Now, the broad category of the another one that we have is nanotechnology. Everyone knows that nanotechnology is basically we are working on the nanoparticles development where we are using some kind of a micrometer 1 to 100 micrometer kind.
Ma'am, am I audible?
Hello,
Yes, ma'am.
ma'am, am I audible?
Yes, ma'am. You can audible. You can continue,
Okay,
ma'am, if you want.
okay, thank you. Okay, nanotechnology, में as we are talking about it, nanotechnology have a vast area as most researchers are working on it right now because nanotechnology is not only a part of a traditional farming, not is a part of urban agriculture, but it's a kind of a concept we are developing some kind of the particles which can be used in a very less volume to have the huge impact on all over the plants, and you can also going to some kind of you're also going to have the impact on it as in a beneficial term, and it is also used can alternative for the synthetic or the bulk fertilizers. For example, if we are applying some kind of a urea, ssp and mop, and we have to apply these kind of the fertilizer in a bulk in a cages. And now in the alternative of these kind of a particles where we are introducing the nanoparticles to make the available the nutrient uh, for the plants. So we are going to use, for example, 19, 19, 19, 18, 18. These are kind of a very small particles where we are going to use it in a grams, and it is going to cover all the plant area, and it is going to be penetrate towards the through the stomata to the plants, and it have a higher impact rather than of the application which is you are applying to the soil, because soil if you are applying some kind of any bulk fertilizer, then it's going to have a depletion of the nutrients on the same time. And the same alternative, if you are using the nanoparticles as a nano fertilizer and any kind of a pesticide, so it have a greater impact because it's directly going to be penetrate within the mechanism of the plant, and then it's going to have a great kind of impact, and you are going to easily combat any kind of a stresses or can manage the diseases at the same time. Now the another thing, apart from the fertilizers, we are also using the nano pesticides. Then we have the another thing that what we are using, that is your uh, nano bioformulations. Then we are also using nano priming in the seeds where we are using the chemicals and the particles that will helps to boost the seed germination. Then we have some kind of the nano edible coatings that we are also using for the post harvest management. See, nanofertilizers, and we are once we are talking about as nanofertilizers are released nutrients slowly and in a controlled manner to the plant, and it also helps to reduces and to improve the absorptions of those nutrients within the plants. That ultimately increases the yield of the vegetables. And once we are talking about it, will also can helps. It also can be helps to maintain the other essential nutrients apart from the major ones that can be easily going to be introduced within the plant. And these nanotechnology can be also helps in the biofortification once we are going for the agronomical approaches. Now, as I told you that nanopesticide can be also used for the disease management. We have some kind of the nanoparticles, for example, the silver and the copper nanoparticles that have antimicrobial properties and can be mostly utilized in vegetables to control the bacterial and fungal infections. For example, for downy mildew, for the powdery mildew in a cucurbits, they are used as a best alternative for the nanopesticide for the disease management. And it's also going to help you to reduce the hazardous effects as you are using it is in a very less quantity and have a greater impact with that's made very small quantity. Now these some kind of the nanopesticides and the insecticide also comes in encapsulated forms, and that is also helps to maintain the targeted pest control, and that have a very minimized kind of environmental impact on it. See, we have also the examples like silver nanoparticles that helps to control the bacterial wilt. As everyone knows, that is a kind of a disease which is very hazardous in the tomatoes. Then we have a nano chitosan that is very effective against the fungal diseases, especially in your green vegetables. Now, the other benefits of using nanopesticides are reduced chemical residues effect, because we have a residual effect of the pesticide and the insecticides on the plant after the application of it. Then we have can helps to maintain the target or the specific pest and the diseases. And especially this is, as I told you, that you are going to use it is in a very less volume, and you are going to have a greater impact on your plants. So it is a kind of eco-friendly approach. Then another thing that is nano sensors, and these nano sensors have a very important kind of the place in a precision farming, where we are using these nano sensor as the detectors and to detect the nutrient deficiency within the soil. Then what kind of stresses are be there in the soil or within the region, and what kind of the diseases, to collect the real time data, so we can plan the layout according to that. And we can also going to have some kinds of sensorized like drones we are using for the application of the fertilizer. Then what kind of a irrigation method, what kind at what time you have to apply the fertilizer, irrigation, and all the package practices has been decided according to that collection of the real time data. And this also helps to reduce the resource wastage, as in the traditional method we are going through the laborious work, we have the expensive kind of the labor, we need a skilled labor for it. Apart from that, we are relying on the bulk fertilizer, on the expensive kind of the other pesticide. And the same time, once we are using the nano sensors, it is going to give you a very quick kind of the instructions. So you're going to have a smart nano sensor that will help to detect all kind of the deficiencies, disease, and how you are going to manage it. So if I am talking quickly for the benefits of it, that is effective efficient use of water and nutrient management you can do, early disease detection you can do, and then ultimately you are going to increase the productivity of it. Another thing is your post-harvest preservation and packaging. As previously I told you, this is also a part of your nano technology, where we are using nano-coated and nano-edible waxes. That is very, very popular nowadays. So nanotechnology not only enhances the quality, maintain the so another thing we have, like the post-harvest management. Then we have the another concept, as I have to conclude the slides, biofortification, as I told you. And biofortification, so we have some kind of the agronomic approaches we are opting for it. Then we have breeding approaches and transgenic. Uh, Agronomic approaches where we are applying the fertilizers that is in the form of the application via through the soil or by the foliar application that will improvise the quality of it. Then these are some kind of examples where we can targeting some kind of a nutrient to improvise and to fortify it within the living plant or the cultivated plant. That is your tomato may, for example, iodine, zinc, selenium, beta carotene, and other kind of the nutrients. Then another, we have a pre breeding objectives for the quality vegetables, and this is also a part of biofortification. And we have some kind of the traditional or the kind of species which is used to improvise. It's like Solanum pimpileni is the species of the tomato that have a high ascorbic acid and can be used for the hybridization and the introduction or selection of the further development of the breeding of the cultivar development or the hybrids. Then genetic modifications, we have some examples of is as the alteration of the starch quality, then have a reduction in anti-nutrient elements in the sweet potato research quality that is going to be improve the starch quality of the plant, that is due to CRISPR, as they are using the editing tool of CRISPR-Cas9, some of talents. Then we have the other examples of the nutrient rich, as I have like VT95, which is developed from HPKAS Almora. Then we have Kasuri Sinduri. Then Kinder Red, that is a red cabbage. And the conclusion of these all the slides, as we are talking about the current status of the research area in vegetable production. See, we have a huge and the vast area, as we have to significantly works on the area, like on the nanoparticles, on the advancements of biotechnology, and then the very important aspect is biotic and abiotic stresses. And they see vegetables are directly related to the population increasing, the health, to maintain the health of that population. So we have to encourage the some kind of a collaboration with the scientists, with the farmers and the policy makers. So this is in for all the topics whatsoever I have discussed over here. If anyone have a question there, so they can ask me.
Actually, ma'am, we have, due to some technical issue, we had made this meeting live on the YouTube only. So
Okay.
participants are not there in the group, this meeting. But yes, if we get any query or anything from participants, we will surely share with you, ma'am.
Okay, thank you so much, ma'am.
So on the behalf of everyone, I just want to say thank you for taking the time to such a, deliver a lecture of insightful lecture on nanotechnology, ma'am.
Thank you, ma'am. Thank you so much.
Thank you so much, ma'am.